So there's a village where the houses are producing garbage. Garbage is there. And there are trucks that can clear the garbage. But every year new houses are built. And before long, it's a big city. Now they're making a lot of garbage. And the trucks, it's a strange country. You don't get more trucks. You still have the 100 trucks you began with. And now the trucks are overloaded, busy with the garbage all the time, overloaded. And when they go on strike, the garbage even goes more. That's like noise, or you add some more garbage until it reaches a threshold of robustness, which is incompatible with life. It's the metaphor of the village. And it decomposes the problem into slow, linear rate of increasing houses, which are garbage production units. We'll talk about what they are in the body. So damage production increases with age, and the damage removal saturates it. It doesn't have infinite capacity, and there is a robustness threshold.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Dr. Gil Blander. Welcome to Longevity by Design, how to live longer, healthier life. We are produced by InstaTracker, your science-based guide to optimize your body from the inside out. Our guest today is Dr. Uri Alon, professor in the Department of Molecular Cell Biology at the Weizmann Institute of Science and pioneer of system biology. Professor Alon is best known for discovering network motifs, the fundamental regulatory circuits of life, and for reshaping how we understand robustness, resilience, and adaptation in biological systems. More recently, his work has challenged long-standing assumption in aging research, suggesting that gene and gene regulatory network play a larger role in healthspan and longevity than previously believed. Uri, welcome to Longevity by Design.
Hi, Gil. Thank you for having me.
Oh, thank you so much. And as we discussed pre-recording, I know you from my days at the Weizmann Institute, and I really appreciate that you came to our podcast. And I also really appreciate that you move into the longevity field, because I think that people like you can help us expedite the discovery, understanding, and hopefully finding tools to allow us to live longer, better. But before we dive in, if you enjoy Longevity by Design, please like, follow, and leave a review on Apple Podcasts, Spotify, or YouTube. It's help others find this science-first conversation. So, Uri, you began your career in physics before becoming one of the founders of systems biology. What drew you into biology?
Well, physics is amazing because you learn to look for simplicity inside complex phenomenon. And that's something I carry with me. It's almost like a disability. I have to look for simplicity. Sometimes I don't find it, but if we do, it's a big gain. But physics, when I was a graduate student, it was mainly about huge things like the sky, or tiny things like subatomic particles. And I was interested in human beings. And so I started looking for other fields where you can use math and to understand it. And a colleague gave me a random book in biology. It was called Recombinant DNA Biology, paperback. And I learned that there's such a thing as DNA replication, and there's something in ribosomes. I had no idea. I said, oh, there must be new laws of physics there. And I was kind of in love. And then I did my postdoc actually working on bacterial chemotaxis on biology, became an experimentalist. And then when I started my own lab, it's kind of physics combined with biology, with medicine and computer science to understand, find some angles of simplicity inside the complexity of biology.
Cool. Thank you for the simple introduction, let's say. And what does a system perspective reveal about a living organism that traditional biology often misses?
I think it kind of asks a new level of questions. So if in molecular biology, the answer is a molecule, a pathway, system biology starts from knowing the molecules in the pathway and asking how they work together to achieve the wonderful miracles of what a cell can do, what an organ can do, what an organism can do. So it's at the level of finding principles, mathematical, mechanistic models to bridge between molecules and behavior.
Yeah. And why it's specifically so important in aging that so far we know that we poorly understand what is going on there.
Aging research, I think, is pre-theory, data rich, and theory poor. I mean, there are lots of theories, quote unquote, like reactive occupant species drive aging or telomeres drive aging. That's not really theories. But I do think that when you see that there are patterns inside aging that unite many different organisms, you understand that there has to be a hierarchy of some processes that are more important than others, and some processes are upstream of others, and you can dare to build a picture that explains the observations. Why is it genetically identical organisms raised in the same conditions die at different times, right? Why is it that the risk of death rises exponentially or power law in so many creatures? Why is it that midlife interventions in animals quickly go to the better survival curve? All these kind of patterns. Why do diseases rise almost the same rate? Very different diseases. Why is there a slowdown at old ages? All these things together constrain you to basically very small number of possible mathematical processes that can explain aging. With those mathematical processes, you could use them as a lens to look for a needle in the haystack. What are the drivers of aging? Because you understand them from that perspective. It's uniting data from epidemiology and medicine with molecular and biochemical, usually fields that are hard to bridge.
Yeah.
Did I answer the question?
Yeah, I think so. And let me say it in my word to be sure that I understood it, and hopefully our listener will understand it. It's basically what you are saying. It's allow you to take a massive amount of data and explain it like what you said at the beginning in the simplest way possible. And if you can explain it in the simplest way possible, most likely that's the explanation. That does make sense?
Well, it's an explanation you can understand, first of all. It's not like a black box.
Yeah.
And then what you do is you try to test and torture your favorite model in all possible ways. And what happened with these models is that they start explaining things that they weren't even meant to explain, like how much do genes affect our lifespan, which we'll discuss, or other questions again. And then you start saying, okay, this is simple. It's simple enough to explain a lot with a little. And that's how you gain more and more confidence. You can never prove it. And also, if it makes you do new experiments, then it's a good model. And so you wanted to make new experiments from which you can learn more about aging, and that's what defines a good model.
Yeah, makes a lot of sense. And following on that, you introduced the concept of network motifs. What are they, and why are they so fundamental to biology?
So this was around 2000, and by that time there were lots of protein discovered in human cells. Let's say tens of thousands, and also a lot of the ways the proteins talk to each other, interact. And when you try to make a picture of that, each protein with an arrow to another protein, you get what's called a fuzzball. It's an ununderstandable mess of information. People were really frustrated because how do you get from this fuzzball to the things that the cells do, and to aging one day, etc. And we looked at it like a graph or a network, which was very unusual in that time. And when we looked at it, we saw that it's actually made of simple recurring patterns, like a feed-forward loop, where a gene activates another gene and also activates a third gene that regulates the second gene. So it's like you activate a gene and its regulator in a kind of a triangle. And then there are 8 kinds with repressions and activations, and you find some of them and not the others. And then they have a function, like they can cause a delay when you turn it on, or no delay when you turn it off. And we got really excited. And then you discovered the same small set of recurring circuits, as we'll call them, in many different cells, many different organisms, from yeast to humans. And they shed light on what the dynamical computation the cell is doing, each time with a different gene that does the biochemistry. And this also became the basis. Once you see circuits, you can think of designing new circuits. That became part of the theoretical basis for synthetic biology, which builds new circuitries inside cells, as we see now in redesigning T cells for cancer, etc., bacteria to make stuff. And it's also showing that evolution produces things that look like engineering, even though engineers learn in their books what are the best circuits. Evolution works by tinkering with found parts with what it has. But it seems like in order to develop a system that can scale up, you have to use a small set of very robust and very simple circuits in order to build up complex functions. Otherwise, they have no chance of working. And it seems like evolution has, quote unquote, rediscovered the same principle that engineers use when they build an iPhone, always using the same simple circuits again and again and again to make memory elements, etc. So this became the language, I would say, one of the languages of understanding the cell on the systems level.
So that's it's interesting, but in a way it's also scary, because if you have like a specific circuit that they are very important, it's let's say that like let's say the power plant in a snowstorm in Boston, and if the power plant not working, then everyone will stay cold. So that's also we can find what are the key let's say processes that we need to be very careful about maintaining, correct? Or
can find
intervening. Yeah.
You can find vulnerabilities, which are concerning for our health, but there are also control points, which are the best points to drug.
Yeah.
For example, it turns out from these network motifs, if you try to build a drug to affect a function, most places you try to intervene are going to be buffered out by the system's incredible robustness. But there are those fragility points, like you say, which are levers that you can actually tune as a physician or as a chemist trying to make a drug. And so the control of biological processes is also great. If you have a blueprint with circuits, you know what to do. Like imagine trying to fix a radio without a circuit.
Yeah.
It just sheds light on the problem.
So if we are now thinking about aging, what you are trying to do in a way is to find those key circuit, and then find how can we tune them up, and hopefully we can increase the healthspan or the lifespan. We'll get to that maybe later, but that's what you are trying to do.
Yeah, you can say, first of all, trying to understand, because I'm into understanding. And for me, it's a joy to figure things out and to find some kind of simple explanation that explains a lot. That's my upbringing. That's why I'm in science. But of course, aging is also personally interesting to me and those people that I love. And I do believe that it can contribute to the search to slow down the core process of aging in order to delay all aging-related diseases together. A very elegant solution for prevention. It is called the geroscience hypothesis, and I think is a revolution in medicine waiting to happen. We can discuss later. That's not the same as extending maximal lifespan.
Yeah,
but we can talk about that.
No, I think that we should discuss it now. So basically, we call it in the aging domain healthspan versus lifespan, and I think that our audience understand it.
Yeah.
Nobody want to live to 500, but be on a bed for 400 years. We want to live to 500, but be alive, ride your bike, climb a mountain, go to your lab and do experiment, have fun with your family.
Society.
Yeah, exactly. So absolutely. So I think that what you are saying makes a lot of sense. So Uri, related to aging, and if we are starting to dive in, so it's often described as accumulation of damage. And your work suggests that something more structure is happening. So how this structure or regulatory network change with age?
Okay. Yeah, so I want to tell you our framework and for the listeners, but I'm not going to use math. I'm going to use a story about a village. The nice thing about it is that I use it also to compute. After the story, we'll be able to understand the difference between damage production, damage removal, and robustness to damage, and noise, and all the things. So there's a village where the houses are producing garbage. Garbage is the damage. And there are trucks that can clear the garbage. But every year, new houses are built. And before long, it's a big city. Now they're making a lot of garbage. And the trucks, it's a strange country. You don't get more trucks. You still have the 100 trucks you began with. And now the trucks are overloaded. They're busy with the garbage all the time, overloaded. And when they go on strike, the garbage even goes more. That's like noise, or you add some more garbage. And there's, until it reaches a threshold of robustness, which is incompatible with life. It's the metaphor of the village. And it kind of decomposes the problem into slow linear rate of increasing houses, which are garbage production units. We'll talk about what they are in the body. So
Yeah.
damage production increases with age, and the damage removal saturates. It doesn't have infinite capacity, and there's a robustness threshold. So this is, I'll say just briefly what we think these are in humans. In each organism, it's different, but we think in humans that there's different candidates, but garbage, we think, is damaged cells and senescent cells that are very inflammatory. And when they build up, they make the whole body full of inflammation and slow down stem cell removal. And the trucks are the immune system that removes those zombie cells. These are NK cells and macrophages, maybe other cell types. And of course, you don't get more of them with age, much more. Not enough. In fact, they get exhausted with age.
Yeah.
And the houses are a little bit of a mystery, but the houses, we think fundamentally, have to stay in the body for all your life. So they can't be a skin cell that's lost after 30 days or a gut cell that's lost after 5 days. So it has to stay in your body for all your life. And there's two kinds. There's permanent cells like neurons, and there's stem cells, which are dividing all the time. And because they're dividing all the time, they accumulate epigenetic changes or chromatin opening changes that are fine for those stem cells, but when they transmit it to their daughter cells, they're trying to divide many times in order to supply this cells in the body. That causes problems, all kind of problems. The double-stranded breaks, telomere shortening, jumping of viruses. The cell tries to repair it, but sometimes it can't, and then it becomes a zombie cell. So those stem cells are like factories that more and more of these altered stem cells arise in your body. So with age, you accumulate factories that produce garbage. DNA repair is inside the houses, and that framework provides the 3Ds of aging: death, disease, and decline. In this little model, the rising of houses with time linearly and having a threshold goes into the exponential rise of risk of death, because you have to throw coins. Those are the noise, and you need to cross the thresholds. You have to be very unlucky when you're young, but much easier when you're old. When garbage builds up, you need fewer coins. So it's an exponential rise of death. Also of disease, because diseases have a lower threshold for diseases, and that explains hundreds of diseases we see from medical records to right. And even decline, the fact that a lot of organ functions decline linearly or gradually with time. All this is explained by this model. So when I saw that the 3Ds are explained, I said, wow. And then it also explains interventions. For example, if you do exercise, what are you affecting? Producing damage, removing. Turns out when you exercise, you increase your threshold, the amount of damage you can stand before you die. For example, before you die, you need to breathe a lot, take a lot of oxygen. So your VO2 max is larger, your vasculature is larger, your mitochondria are larger. So having more a ready body allows you to withstand whatever is going to kill you. So, and then mathematically what happens is and I think everything that happened in the last 150 years is increasing our robustness. We still don't know medically how to reduce damage production and removal. We'll talk about that later, like Senolytics and mTOR and stuff like that. Everything we know, all the lifestyle interventions change the robustness, make you more able to withstand damage. And the effect that has is to increase the median lifespan, but not the maximum lifespan. So that mathematically can explain why in the last 200 years, our average lifespan has doubled, but our maximum lifespan is still a wall of 120. And when you increase robustness, you prevent early deaths, but late deaths because damage accelerates so much. And you basically get this rectangular curves that are pushed up against the 120 wall more and more. In order to move that wall, this model tells you what you need to do. For instance, you have to address DNA damage repair, chromatin states, repair, let's say mTOR that increases repair, all kind of repair. That's in the houses. You have to slow down the houses. Or add drugs, like senolytic drugs that kill senescent cells, or upgrade your immune system. Okay, and this provides a framework to analyze what interventions do, what interventions we need. If we think about Healthspan, for example, some interventions like increasing robustness compress morbidity, or they add years without years of sickness. But on the other hand, things like mTOR inhibitors, which affect the houses, they'll stretch lifespan, but also the sick years proportionally. So if you double your lifespan, you'll double. That's not good. So you have to add, because basically slowing down the houses is like moving time more slowly.
Yeah.
And so in order if you want to do that, you have to add another intervention, like on the trucks or in the threshold, in order to make it steeper too, in order to compress morbidity. So all these insights from such a simple model, and then you can check that in animals, and it works exactly like I said. The interventions that just stretch out time, extend the sick span in proportion, and interventions that steepen the survival curve, compress morbidity in animals. So this simple model is like a framework for which you can run in your head what'll happen if I do this, which two interventions are good to add if they work on trucks and houses. It's good to add them. Sleep, we think, works on noise, makes noise more or less. So sleep and exercise should be additive. And this is the whole world of like a playground we're playing with to explore aging. And of course, this also acts as a lens. We can go to model organisms and figure out what exactly is in the houses out of all the, let's say, chromatin changes. So like we can go to yeast deletion libraries collected by Brian Kennedy, and each strain has a survival curve, and we can look at the shape. Is it steep? Is it not? And understand, does it work on the houses or the trucks? And build a genetic pathways for aging, which are very specific. Like we see in yeast, it's histone 4K16 acetylation in repetitive areas. It opens up the chromatin, causing transcription, causing R loops, causing collisions with replication fork, causing double-stranded break with an attempt for homologous recombination that doesn't work, or the lysosome becomes alkaline, making cysteine in the cytoplasm. All these very, very specific, molecular specific. So that's also unexpected for me. From a mathematical model, to get down to H4K16 acetylation in line 1 elements or whatever is amazing, is unexpected. But I think it gives you like a very sharp lens for new experiments, which, for example, we're doing in human blood on those acetylation in order to test the hypothesis, going all the way from yeast to human. So for me, it's a super exciting adventure.
Yeah. And for our audience, so basically, what you are saying is we have a village with houses, garbage, and garbage trucks, and that can explain the aging phenomena in a high level. But then you can build a sub-model below that, like what you explained. And for the audience who doesn't know Brian Kennedy, even so that he was a guest in this podcast before, is a scientist that develop a lot of interesting science, but part of it is screen yeast library with thousands of different deletion or removing of a specific gene from the yeast, and look at the lifespan of them. So what we can do now, we can put it on top of this model and try to understand, okay, this mutation, specific mutation, is it influencing the truck, the garbage, or the houses?
You got it. You see, the simple model, you can now compute with it in your head. It's like,
yeah.
But we do the math, but we also use models like that to get intuition.
Yeah. So I think that what is, I would say, brilliant in your model is that you can go zoom in and zoom out and understand it. And it's going back to the point that you described before, that 20 years ago when you came to the Weizmann Institute, or maybe even more, people came with interaction of a protein, and basically it was a big ball that nobody could understand. But what you are doing, you are basically have layers. So you can look at it in a 30,000 foot view, 10,000 foot view, or 1 meter foot.
That's right. It's like terminal levels of organization, and each one gives you a different answer, a different superpower.
Yeah. No, that's a very clear explanation, at least for me, and I hope that both of us communicated good enough for our audience. I know that it's tough, aging is tough, and but you are a very good teacher, so I hope that they got it.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to Healthspan optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start Inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
So my next question is, okay, so we have the houses, the garbage, and the garbage truck. What if you look at a long-lived people, let's say centenarian versus someone normal that die at the age of 80?
Yep.
What do you see in this model? What is the difference between?
By the way, if people are interested, Gil is going to put links to a free YouTube online course that I taught at Weizmann. I also play guitar there, and I explain this in depth over 12 lectures.
And Uri, maybe, so how can they find it if they want to, if they're just listening?
Type Uri Alon and systems aging and go to YouTube.
Okay, excellent. Yeah, and I want to say that I haven't had the chance to listen to all of it because it's a few different courses and each of them is an hour, but I listened to the first couple of classes so far, and it's amazing. So I really recommend everyone to watch it.
Yeah, it's on the whiteboard and everything. So
Yeah.
research on centenarians is fascinating. And because there are so few centenarians, you can find the common variants. And the common variants, I would say, just as the first order, you're dealt a lucky hand with variants that are found in the population that protect against diseases of old age.
Mhm.
Each centenary deals a different lucky hand, but it hits the main ones: cardiovascular diseases, Alzheimer, and diabetes, sometimes genes that make you smoke less addicted to smoking. And that's great because if you have this garbage accumulating, and diseases are like tripwires, they're below the top threshold. You're susceptible to, let's say, cancer, garbage will cross that tripwire. These genes remove the tripwires, and you can have more chance to reach 100 without dying from those diseases. More recently, but you don't find genes for DNA repair so much because those variants are extremely rare, and you only have a few thousand centenarians. But more recently, there was a breakthrough looking at a situation of aging, which is much more common: menopause. Menopause happens to all females around age 50. And there, researchers like Anna Murray in England took many women and looked for mutations that delay menopause or make it early, which we know is linked to all-cause mortality. It's probably an aging period. And there she found all the classic genes we know from yeast, mice. She found DNA repair, RECQ helicases, really the classic aging genes. So I think there is a big scope to look for. Of course, these are much rarer that can shift our lifespan by affecting the houses a little bit. In other words, the centenarians have threshold genes. They're more robust to damage. They don't get the disease at the same amount of damage. They have threshold genes. But we also have house genes that slow down the houses, tiny little amounts, and each one of us has a set of them. And that's going to determine our genetic potential. By the way, diseases of accelerated aging, the tragic diseases where kids die, let's say age 14, looking like an old person with white hair and heart failure, or at age 40, those diseases are also in very specific DNA repair. They're very similar to what I find in menopause. So you can breaking a single gene can accelerate aging. Luckily, very few genes, by the way. And by the way, maybe I should talk about our discovery about how important genes are at this point.
Yeah, sure.
So in your reading textbooks, in books on aging, they say that the life expectancy is only 20% heritable. And recent papers using family trees in the internet going back 300 years say it's even less than that. Maybe it's only 10% heritable. And that creates a kind of dissonance, because if you're working on aging genes, how can they be only 20% heritable? Or you can say, oh, great, then it's exercise. It'll exercise and reach 120, no problem. We revisited those twin studies that had the classic heritability of 20%. And the thing is, those twins were born in 1870 to 1900, Danish twins, Swedish twins. And back then, people died not from aging, but from, in young age, a lot of times from pneumonia or from having a fall and having sepsis or from diseases like that. Much, much more than we do today. So basically, because people died so early, you couldn't see the genes shine out. Who cares how long your parents live? And what we could do mathematically, we don't have the cause of death, but we could mathematically correct for that, what's called extrinsic mortality, and discover that it's actually 50% genetic. If they said, wait, maybe that's only our math model. So we got new data from Sarah Haig from Sweden about twins that were born later, 1930. And when you do the regular heritability studies, you see that they have 40% heritability. And when we correct for that extrinsic mortality, again, 50%. So we're convinced that even though in 1870 genes weren't that important because you die, it's true, it's low, it's 20% heritability in 1870. Today, it's probably 50% heritability. Even medicine with statins and stuff, those twins even saw statins. So medicine hasn't affected that 50% yet. But maybe in the future, we'll have medicine that can counteract our bad genes.
Yeah.
And then it'll be less genetic again. But right now, it's half and half. But so some people say, wait, does that mean I need to stop exercising? And say, no. I still swim and eat my salad and everything, because if your genes say you're going to make it to 80, if you have bad health habits, you can easily shorten your life by 15 years. No problem. But if you have good health habits, you can probably extend them 5 to 8 years beyond your genetic potential. But the most important thing about those lifestyle factors is that they're good for you right now. Exercise is good for your mood. Sleep is good. Nourishment is good. Connections and friends is, of course, makes life worth living. And so it's both for extending your health span and also for living well right now. But this 50-50, I think, is really important to be realistic. Nothing you can do, we know right now, can get you from 80 to 100. Not exercise, not sleep. And also all those lifestyle interventions have diminishing returns. I mean, you exercise for 150 minutes, you get 3 years, then another 150 minutes, you get another year, another 150 minutes a week, you get a third of a year. So not to be extreme, but yes, to combine things optimally. For instance, we think that sleep that affects noise and exercise that affects the threshold are great to combine because they work on different things, right? If you work on different things, it's additive. If you work on the same thing, 2 different interventions of robustness, you get diminishing returns. So this 50% also is also very encouraging for scientists, because I always say, oh, genes do matter. And now if we can find genes, we can find, what's the pathways, and then we can turn that into pills.
Yeah.
And that's also very hopeful for the 50% that is genetic.
Yeah. So that's fascinating. And if you, let's start with one follow-up point. I know a lot of people that I know that I'm telling them, hey, exercise is good and eat well is good and all of that. And they're always coming and telling me, hey, I know the guy that was a marathon runner and he died from heart attack. So my response to those people is, yeah, there are some that are unlucky, but that's 1 in a million. But think about, it's most likely won't happen to you if you run a marathon that you will have an heart attack. So don't take that as a negative sign. Yeah, exercise is good. The second point that I wanted to discuss with you and try to understand, how do you explain that before, what was it? Was it a problem with the hypothesis or problem with analyzing the data? Because until your paper that actually came out a few weeks ago, yeah, and everyone believed that genetic is 20%. It was an enigma. How come those centenarians live so long? Because genetic is the effect is so low. So how do you explain it that nobody found those background issues?
I think the previous researchers got the correct answer, but not for modern times. So the researchers got 10% for 300 years ago. That's right. The extrinsic mortality made it so that heritability is almost nothing, because so they got the right answer, but it's not relevant for us right now. And the problem was that there was a lack of mathematical tools to do it right, to get rid of the extrinsic deaths, which are, to first approximation, not related to genetics that much, subtracted from the intrinsic deaths. And you need to have mathematical models, and we used several different models and we got the same results. So you need to do it right. And the people who did the twin studies, they're used to studying things like height or kidney function, which you just do how much are twins correlated with each other to get the heritability. they're not used to this weird trait of life expectancy, where if you die young, you're censoring the population and you're actually increasing the additive genetic variance. So it's like a very strange genetic point of view from their background. And now we're going to have new data on 10,000 Swedish twins where we have detailed life history, exercise, and we're going to figure out in the 50% how much is attributable to each kind of lifestyle factor and their combinations, and how much is attributable to sheer biological noise, because that's also a component. The trucks go on strike. They go on strike because each night is different and trucks work at night. That's circadian variability. Because of infections, because of stress, there's some kind of noise that's irreducible. In my opinion, it'll end up to be about half, the 50% will be half luck and half under our control. And it'll be very interesting to see what's the main levers and what are actually not so important levers. So the twins are very valuable. Did I answer your question?
Yeah, no, it's great. So basically what you are saying, as of today, based on the data, the best data that you have, 50% or so is genetics. And your assumption is like 25% is a luck, like, I don't know, you cross the road and the truck eat you. It's
not that. That's extrinsic mortality. I mean, even intrinsic mortality.
What is luck? So what is luck?
What is luck? So in our model of the houses, trucks, and robustness, I told you the trucks go on strike once in a while.
Okay.
The whole immune system is some nights is better, some nights is worse. So the garbage goes up and down like that. That's biological noise.
Okay, so cyclic behavior of your immune system or your body and then at the same time you have like a attack of a virus.
Yeah, so it's not exactly cyclic. That's the problem. In biology, nothing is really like a clock. So one night your circadian rhythm, one night is better, one night is worse. Some of it has to do with your behavior. So people with regular sleep have a little bit longer life, but very steep survival curve. So they all die at the same time as their friends, you can say, which is great, because when you're old and you lose a friend, you lose the whole world. So we see from our model, that means that people with regular sleep, not only long sleep, but regular sleep, they reduce the noise. So we know the noise comes from something with irregular lifestyle, like monks and nuns with very regular lifestyle have really, really sharp survival curves. They're very, very, they live a little bit longer, but very, very equitable life, life expectancy. Yeah. And that's, we think, in our models, that's why we have this model, like we can separate it out. That's the noise. So there's a biological noise. Even if you are a monk or a nun, you'll still have, or if you want it, if you have genetically identical worms grown on the plate, in the same plate, they still die at different times.
Yeah.
Beyond genes and environment. So that's one part. And then there's environment, which has to do with our stress, with our exercise, with our nutrition, with our sleep, with our friends, with our sense of meaning, with exposure to nature, music, being around green spaces, being around blue spaces, all those good things. Yeah. Of course, avoiding excesses, smoking, alcohol, pollutants, toxins, all this thing, we think accounts for 25% of the variation. That's just an order of magnitude, okay? And even we have even twins with the same genes, but raised apart. That data can tell you how much is in the shared environment of the twins grown together. And the shared environment of the twins grown together is tiny fraction. So it's something else. It's very, very interesting to find out. I think that will be the frontier. And I think it's...
But talking about those twins that were separated, they usually twins live together, let's say until the age of 18. So it could be that the effect. So it could be that the effect of the environment is later or earlier, and maybe you can research that as well. I assume.
Yeah, that's what I mean. The shared environment component is very small. It's less than there.
Exactly.
But then there's twins can be different. You know, one can exercise, the other one not, etc. Also, the way our genes are converted into our body. So you know the way the genes convert into the brain wiring. Or I love to look at my hands. Here are the veins on my wrists, the blue veins you can see. Everyone can look at your wrists. You can see the blue veins. They're different on the right and left hand. That, even though it's the same genes in the same environment. Why is that? Because the genes turn into a body through a stochastic process. The blood vessels are looking for places without oxygen. And that gives a different solution on your right hand and your left hand. That's why genes aren't everything. They make a different body in those different twins that can also affect how you interact with your environment and stuff. So some of it is that the way you develop. Some of it is biological noise is going to be there anyway, that you're not going to be able to reduce, I think, unless we have very sophisticated technology that can measure our immune system ability to remove zombie cells and intervene day by day. Maybe that's also possible. And but I think so this noise is also a fascinating concept. It's always been there in aging research, and I think now we have a handle on it.
Yeah. So a follow-up question: if you have a magic wand and you will do the same study in 100 years, do you think that it will be still 50% genetic, or it will be more?
No, I think it will be less, because I think what happens
It will
there,
be less.
Yeah, because there what will happen is we'll know if you have dangerous genes, and we'll know how to treat them. Right now, if we have familial high LDL cholesterol or LP little a, or those genes that you can test, I can give you statins.
Yeah.
And you won't get the heart attack that you would have gotten at 50. You won't. You won't get a bypass surgery. And basically, you've neutralized that gene's effect on your life expectancy. So in my opinion, hopefully, we'll have a very good understanding of the relation between the genes that promote long life and shorten long life. And we'll have the medicines or the approaches to neutralize the bad gene. That's another thing, I think. If 50% of lifespan in your genes, it's not impossible to think we'll have a polygenic score for aging, which is the way from our genome to understand at least that 50%, so you can plan. You can plan your life. You know your vulnerabilities. That's for the future. And it just, this finding, by the way, I was very surprised. We had two papers at the same time on cancer and on this. And this one caught public attention, like it was in all over the press, etc., because I think people reflect their fears and desires on this notion of heritability and lifespan. Some people tell me, oh, now I feel less guilty for not exercising, and also doubly happy because I can blame my parents. All kind of funny things.
That's not the outcome that we want.
No, that's not the outcome that we want. But it kind of, I think it gives a good proportion, really quantitative, what you can do with the levers we have right now, where we should concentrate, what we should concentrate about genes, how you can maybe think about your lifespan and plan ahead, and also what you said in the future, how we can discount the bad genes using pharmacology or technology. And
so going back to, I'm still in genetics. Sorry that I'm double clicking onto all the time because it's very interesting. So there are what we know so far about Centenarian is that there is not, as we said before, there is no one gene that all of them have a issue with or a positive effect. It's a lot of genes. Now, when we started the discussion with you earlier today, you said that basically there are a circuit or nodes that are major nodes that are basically the key places that control something. Yeah. So have we found already those nodes for Centenarian or for longevity, or we are still looking for them?
So in Centenarians, like I said, you get a hand of lucky cards, but it's a different hand every time.
Exactly.
What's very clear is those rare mutations that make you age much faster. Those are very few. So we'll know that just like you said, this is the hub that's so fragile. If you disrupt chromatin structure inside the cell with MNA mutation, you age 7 times faster. And if you disrupt REQ helicases that remove the stickiness of DNA that gets opened by R loops, you age twice faster, et cetera, et cetera, et cetera. We also know that there's 2 clocks in the body, one for the body, one for the brain. We know that because the diseases of accelerated aging spare the brain. You get old, you have cancer, you have heart disease, but the brain is okay. On the other hand, so that's a different clock for the brain. Also, Alzheimer's and Parkinson's rise much faster than all the other diseases. They have a different clock. What's in the brain? These rare genetic diseases that affect the lysosome or the mitochondria, that is the cell's waste basket and the cell's power factory, they ravage the brain. So, and from our work in yeast, we know that that's the second clock. It has to do with this waste basket, the lysosomes getting alkalinized and making all kinds of aggregates. And so that's very interesting that we have 2 different clocks and 2 sets of crucial genes. And I think if we intervene in the very early stages, these DNA repair in chromatin parts or in the lysosome mitochondrial parts, that's the secret for, that's one way to think about a lever that can slow down the houses. By the way, when we look in evolution at different animals, mice that live 2 years, elephants that live 80 years, whales that live 200 years, what evolution tunes between different animals is primarily slower houses to live longer. And that slower houses can be achieved by repair inside the houses, by more recycling, by more reuse, etc. And each animal is different. So we know that's the most important lever in order to slow down time, basically, in all aspects of it. Of course, also removing trucks, increasing trucks will work, etc. So I think we know already the main pathways that we need to target, from looking at long-lived animals, looking at diseases of accelerated aging, looking at accelerated menopause, looking at different organisms. We have that set, and we have it even in order. It'll be chromatin before DNA breaks, and it'll be lysosome before mitochondria. We have like an organized structure where to intervene. And now it's a technological question: how can we do that? And there's of course different approaches we can discuss, like partial reprogramming that aims to clear the epigenetic marks. That will be a classic slowing down the houses.
Yeah.
Where as opposed to Senolytics that destroy senescent cells, will be a classic adding more trucks.
Yeah. It's adding more truck or removing the garbage?
Yeah, that's what same thing.
Yeah, okay.
That's what the truck, it's not adding more trucks, it's giving a boost to the trucks by removing garbage. It's not
removing garbage.
We add more and better NK cells and macrophages or something. I don't know if that's possible. But or CAR T cells, there's even work on CAR T cells to target senescent cells. So adding our own, taking out our T cells, engineering them to recognize the zombie cells, putting them back in mice seems to eliminate them. Also, PD-L1 inhibitor seems to eliminate them. So there might be approaches to add trucks, or like you said, to remove garbage to help the truck. That, by the way, unburdens the trucks and makes them free. What do those trucks do? It's not only fight zombie cells. They're the same trucks that are first responders to viruses. They're also the first trucks that are first responders to cancer. Part of the reason why infection cancer is so deadly when you're old is the trucks are busy with the zombie cells. That's another thing to consider.
So maybe my view of that, maybe instead of calling it a garbage truck, maybe call it a maintenance crew or something like that.
Yes.
Because as you said, they are doing the electrician and they are cleaning your house and making it.
Yeah, they're multitaskers actually, like first responders or something.
Exactly. Maybe first. No, it makes sense. So let's talk for a second about epigenetic reprogramming and Senolytics. In your opinion, which of them will make a bigger effect on longevity when we'll get to the point?
First of all, they'll make a different effect. One will slow down the houses, which is going to extend our median and maximum lifespan by the same amount. But on the other hand, we'll stretch out time. So it'll also stretch out the time that we're sick, which is bad.
Yeah.
On the other hand, Senolytics remove garbage, and they're going to increase our median lifespan and by more than they increase the maximum lifespan, but they're going to compress morbidity. And we also know that combining them is going to give you the best of both worlds. Expanded maximum lifespan and compressed morbidity. We know that from experiments in mice, which rapamycin that slows down time, and on the other hand, things like diabetes drugs improve the vasculature, and they like build more roads that preserve the roads for the trucks. So they're like trucks. Yeah. There's several drugs like that. And we also have experiments on their combination, and it just sits right where the model predicts. So they'll do two different things. That's part of the importance to understand that there's production, removal, and threshold. And then, of course, you can increase the threshold and get another and reduce the noise, and altogether get a very effective. But in terms of technological closeness, there's no doubt that Senolytics are much closer to, easier to imagine than partial reprogramming. I mean, in the next 10 years, for example, because of the technological hurdles you need for partial reprogramming. Unless, of course, there's a breakthrough. But Senolytics are basically, some of them are repurposed drugs for other purposes. And by the way, I think Senolytics are going to be much easier than finding cures for cancer, because cancer cells mutate and divide and evade and make resistance. Senescent cells just sit there. Actually, that's the definition. They stop dividing. And there's lots of levers where you can attack them and kill them. You can unleash the immune system on them. So I think that problem is going to be solved. And I think maybe we'll have different senolytics for different people. We'll find out that senescent cells are very heterogeneous and each organ is a different one. And we'll find out they're only part of the problem. Like I said, there's two clocks. If we remove even one set of houses, garbage and trucks, we'll find the next one. The next one will be a brain degeneration. And after we remove that, we'll find the next one. Who knows what it is? Maybe cross-linking of the ECM, or maybe it's DNA mutations, which we know don't drive aging, but maybe they do at age 200. We'll have this like an onion. We'll have layers of houses, garbage, and trucks we'll need to address.
Yeah. So other than those two, and by the way, I agree with you 100% that I think that those are the two leaders in extending lifespan, basically epigenetic reprogramming and senolytic. What else exciting you about a new technology or old technology like rapamycin and metformin? What is the other candidates for that?
Yeah, rapamycin. I think I'm very looking at a trial by Yusin Su about rapamycin and menopause, because as I said, I believe that menopause is the marker for aging. And in the initial results, she saw that rapamycin 5 milligrams per week delays menopause. And when that gets to phase 2, I think it will be our first real clinical trial on an aspect of aging I'm very excited about. But I don't take rapamycin myself. Of course, I think it's an interesting debate. But the other technologies that I'm excited about include, first of all, I think there's a huge scope for engineering, like exoskeletons, robots that can catch us if we fall, help us, keep us from being lonely, pumps that measure our important physiological variables, just like insulin pumps do, and close the feedback loop on the major variables that are critical for life. That all those will increase critical. Will increase our robustness. We can stand. It's like having your own personal emergency room. You'll be able to survive more damage. Like that.
So they basically will increase our health span, but not lifespan, correct?
Yes, good, exactly. They'll increase our health span, our median lifespan, but they won't move the wall.
Yeah.
Yes, exactly right. You got it exactly. Okay. So then, in order to reduce damage and reduce houses, I think that if we know that it's H4K16 acetylation and line 1 elements, suppose we know part of the program, it's not impossible to imagine drugs where you add a DNA binding to those repetitive elements, let's say with CRISPR, with a deacetylase. So you can imagine going after the specific epigenetic modifications and close chromatin in a way that's, you could just give a pulse of that or something like that. So reprogramming is very, very amazing, does a lot of things, but a more targeted approach for reprogramming, aging-related things, will have less chance of side effects, I would say. So that direction. And in the world of senolytics, I think reprogramming and our own NK cells and macrophages, taking them out and adding them back in as senescent cell killing machines, our own immune system, I think you can't beat that. Or using inhibiting checkpoints that are used by senescent cells in the old but not in the young. Those are very important senolytic directions, I think.
Well, what about GLP-1? Do you see it as a longevity drug?
Yeah, so GLP-1 is an amazing game changer for obesity. And obesity is upstream of cancer, metabolic disease, neurodegeneration, heart disease. So reducing that is going to, again, increase the robustness threshold, in my opinion. Whether it has also senolytic activities or something like that, I'm not sure has been determined enough. Yeah. It has great effects on the heart that are not independent of its weight effect. So as far as I can tell, it's a robustness raising intervention. Maybe some other things like SGLT2 inhibitors, which work on the kidney and make it secrete more glucose, will also protect the heart. Maybe those have some evidence for senolytic activity or something. But again, we need to target the houses and the trucks in order to make a real dent or push that wall of 120. Did I answer the question?
Yeah. You definitely answer it, and it sounds like that what we see today is that if we look, I don't know, 10, 20 years in the horizon, the two most promising treatment are senolytics and basically
Well, I
epigenetic.
want to add one more thing. From the mice work in ITP, you see that there's another class that I find promising because they work on the trucks. They provide roads for the trucks. And that is medicine that helps you protect your microvasculature as you age. So our body is filled with a network of blood vessels that give us oxygen and remove waste. And they're tiny ones. They're supposed to reach everywhere in our body. And that's also how the immune system comes in to remove the zombie cells. They have to go somewhere. They go on the roads. The roads are the blood vessels. And an amazing experiment here in Israel showed that if you express the hormone that encourages blood vessels just a little bit from the liver in mice, I mean, the researcher did that for other purposes. And then mice lived 2.5 years. After 4 years, they called her and says, what about the mice you left here in the animal house? They're still so young mice. So this is the largest intervention, I think, in mice, a 40% extension of median and maximum lifespan are these VEGF, these mice with good vasculature. And then if you look at the data, you see the truck-like genes in our approach, these genes that look like, or the drugs that looks like they change survival in a truck-like manner, that is to say, changing median and maximum lifespan in a particular pattern, they're all about vasculature. There are carbos, which stops glucose from going into blood. Glucose is what damages blood vessels. There are SGLT2 inhibitors, which do the same. There are blood pressure drugs. So we think that in humans, and even more than in mice, protecting the vasculature might be a way to help remove the garbage. And therefore, it's not a threat. So I'm waiting to have diabetes so I can be prescribe SGLT2 inhibitors. I think that has a good chance of being I'm joking, of course, but that has a good chance of being maybe a senolytic, maybe a blood vessel enhancer, and there's lots of drugs to do that, and that's another optimistic path in my opinion.
So in a way, now going back to your model,
Yeah.
For the houses is the epigenetic reprogramming.
Yeah.
For the garbage is basically the senolytics,
Yeah.
and for the trucks is the VEGF, correct?
No, I think the VEGF will actually for the trucks. For the trucks, exactly.
Yeah.
And then the
So
threshold
basically,
got it.
basically you have you describe very nicely one tool that might help us to improve them for each of the
exactly.
3 different building block of your model.
And that'll add up, both in our model and in experiments. So
and then I assume that those are the first one. I assume that if we'll talk again in 5 years, there will be 10 or different options for each of those.
I agree with you. I think we're just scratching the surface. If you compare cancer research, which is the most advanced field now, to this kind of research, we're still 100-fold less. But even if we take 10% of that talent, I think we'll crack the problem because, as I said, those problems are much, much more naive than cancer because cancer is a cell designed to evade you. It's designed to mutate. It's designed to migrate. It's designed to survive. These are old cells. They say, leave me alone. And they're sitting ducks, basically. That's my view.
Yeah. No, that's great. I think that we summarize it well. So I want to do a quick fire round, and let's start by asking you one myth about aging you most likely to.
Say again the question. I didn't.
One myth about aging you most like to correct.
Genes determine half of your lifespan, and exercising to extreme, etc., won't get you to 100 if you encode in your genes as 80.
Yeah. Okay, that's good. One systems principle everyone should understand about health.
That we need to think about increasing our capacity to withstand damage separately from reducing the way our body makes and removes garbage, damage. And
okay, And one longevity intervention people overestimate.
Overestimate?
Yes.
I'm guessing it would be maybe everything that has to do with transfusing blood from young to old, which I think, parabiosis experiments we can imagine are sharing trucks between a young village and an old city.
Yeah. And also, what you're saying, you suddenly have a newer trucks and it's easier for them to remove the garbage from the old. Yeah, makes sense. One signal that best predict long-term resilience.
So from what I know, it's your VO2 max, your grip strength. I think it's also, do you have something to wake up for in the morning?
Yeah. So basically, a vision. Yeah. No, I understand. Makes sense. So my last question: if the listener remember just one idea from your work on system biology and aging, what should it be?
Tell me, Gil, what do you think? You've heard me.
Yeah. Okay, that's not fair, but I will do that. In my opinion, is what you showed to us is that aging can be looked at as a equation in a way of equation. And when we do that and we simplified it to that level, it's much easier for us to understand it, understand it better, and after that we can understand what are the key points of a breakout and what are the key points of a potential good future, and based on that maybe develop a new drugs or understand the current drug and or the current intervention. It doesn't have to be a drug, it can be exercise or I don't know, instead of doing maybe swimming in only in the left hand is better for you. Something like we cannot know right now, but based on if you basically make equation, you can start debugging them and understand what is going on. That's how I see it.
That's what I would say. Is that aging isn't random or chaotic. There is an equation that gives you at a high level those balance between production, removal, threshold, that helps you understand your body, understand how to read the news, think about height, and helps you be the best steward of your own behavior and body and those around you.
Yeah.
Aging has a logic. Knowing that logic is essential for us to navigate now.
Yeah, and I think that again, if I'm trying to take out the key point for me is what you said, that right now we think that aging is a 50% genetics, 25% stochastic, and 25%, let's say, environment and lifestyle. So I want all the audience to take out of it. You still can control 25% of it. And what Uri said before, with the advance in medicine and science and all of that, the 50% of the genetics will get smaller because we will have intervention that we can basically erase it.
Correct.
Yeah. Okay. Good. So Uri, thank you for illuminating conversation. Your work showed that longevity isn't about chasing isolated fix. It's about preserving the structure, flexibility, and resilience of a system that keep us healthy over time. And to our listener, if you enjoyed this episode of Longevity by Design, please like, follow, and leave a review on Apple Podcasts, Spotify, and YouTube. It's help others discover the science first conversation. Thanks for tuning in to Longevity by Design, produced by Inside Tracker. We'll see you next time.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.